Alright, so for our next section, we're going to start up with chemical reactions and get a sense for how some of these bonds begin to either break or form in the first place, or both. So one thing to remember is that when you look at a chemical reaction, the atoms actually can give up electrons, accept electrons, share electrons. They can partner with different other atoms, different elements, but the identity of elements does not change throughout the course of the reaction. In other words, if you start with carbon as a reactant, you better end with carbon somewhere as one of the products, right? We're not changing lead into gold. We're not changing carbon into nitrogen. We're not doing those sorts of things. We're only changing who the elements are paired with, not what they actually are. That's a really key thing to remember. Right? Electrons can move around, protons don't. Proton number is a fixed aspect of a particular element. Atoms do not give up their proton numbers or change their proton numbers in nature anyway. Okay, so the reactants are the molecules you start with. These are like the raw ingredients. They are the beginnings of the chemical reaction, and usually they're going to be on the left side of an arrow. Here, what you start with. The products are what you get, and so this is the resulting end of the chemical reaction. On the right side of the arrow, this is what results from the reaction itself. So reactants are turned into products. All right, so if you take something like glucose and you add in some oxygen, those are your reactants. And if you then get carbon dioxide and water vapor, those are your products. That, by the way, is a very generalized formula for something like aerobic cellular respiration, but just using that as an illustration to show what reactants are turned into products. If we look at the reaction for photosynthesis, we have on the left side of the arrow the reactants, which are water and carbon dioxide. If we throw in some sunlight, what we get then are glucose and oxygen gas, which are our two products. So these two reactants are turned into these two products. And again, the identities do not change. Over here we see 6 carbons, over here we see 6 carbons. Over here we have 6 times 2, which is 12 hydrogens. Over here we have 12 hydrogens. And over here we have 6 times 1 oxygens, plus 6 times 2, 12 oxygens, 18 total. Here we have 6 times 2 is 12, plus 6 more is 18 total. We've not changed the identity of the elements involved, we've just changed who they're partnered with throughout the course of this reaction. So chemical reactions can be influenced, rates can be influenced by a number of things. Temperature is a big one. So if you go into a chemistry lab, one of the things you'll see is those guys are always using Bunsen burners and hot plates, they're always heating things up and cooking things, and that makes the chemical reaction go faster. And so the reason for that is that by increasing the temperature of the reactants, you increase the chances of those reactants running into each other, exchanging electrons, giving up electrons, accepting electrons, reacting, and then therefore giving up product. Another factor which can influence rate of reaction is how much reactant you have and how much product you have. Generally speaking, the more reactant you have available, the more products you can make. And generally speaking, the more products you have, the harder it is to make more products because they can actually interfere with the actual making more products throughout further chemical reactions. And then a very, very interesting and important way of affecting chemical reactions rate is by the use of catalysts. Catalysts are chemicals which are able to speed up chemical reactions, often by millions or even hundreds of millions of times. In the case of living organisms, the catalysts we are most commonly going to be seen using are enzymes. So enzymes are biological catalysts that happen to be proteins, and they're going to speed up chemical reactions enormously, so much so that if we didn't have the enzymes, reactions actually wouldn't happen in our bodies at a fast enough rate to support us as being living things. And a lot of reactions are reversible. We're going to see this when we look at the production of ATP. So if we take something called ADP and add a phosphate group to it, we create what's called ATP. But just as quickly, if not more quickly, that ATP can break back down again into ADP and phosphate. So these reactions can go in both directions. They can go left to right, they can go right to left in many cases. Right, so our next section is going to be look at some of the properties of water and why water is so important to supporting life on the earth. Right, so some of the properties that water has that many other substances do not have is cohesion. Cohesion is the ability of water molecules to stick to each other, and the reason they do that is because of the fact that water is a polar molecule, and for that reason, water can form hydrogen bonds with itself. So in other words, the partially negative end of an oxygen atom, or the partially negative end of the water molecule, which is an oxygen atom, has a slight negative charge, and the partial plus charge on the hydrogen end of the water molecule creates the potential to bond between the oxygen of one molecule and the hydrogen of another molecule. So that the hydrogen bonding between water molecules allows them to stick together and create phenomena like surface tension, where very small objects can actually rest on the surface of a body of water without breaking through because the water molecules are sticking to one another. And we'll see a picture of that here in a second, which will make it more easy to visualize, I think. There's also going to be what we call adhesion. Water molecules will also hydrogen bond to any other polar molecule. So if you have a water bound to cellulose, for example, it will actually stick to it. And cellulose is important. It's the inside of what make up the water transport tubes of plants. And because water is adhesive to cellulose, which is the inside of those tubes, it's able to climb the walls of those tubes. And that's a very important factor when it comes into plants transporting water, especially. All right, so this one up here happens to be cohesion, where we have two water molecules sticking to each other. We have the partial plus on an H, we have the partial minus on an oxygen, plus attracts minus, and we get this hydrogen bond forming between the two of them, linking them together. So again, this would be cohesion. Adhesions, where we have, again, we have some other polar molecule. This one happens to be butanol, where we have a partial minus on an oxygen, partial plus on hydrogen, partial minus on oxygen, partial pluses on hydrogens. This partial plus lines up with that partial minus. They stick together through a hydrogen bond, and there's an attraction there. So that would be adhesion. Right? So cohesion produces properties in water like surface tension I mentioned a second ago, where this water strider can actually walk on the surface of the water. You see its legs actually pushing in on the surface. It's got a mass to it, so it's exerting a force on the surface of that water, but not enough force to actually break through, because water has this cohesive property. The water molecules stick together, and that keeps the water strider on the surface of the water. This one over here is adhesion, where we have water molecules sticking to the sides of a glass capillary tube. Glass also happens to be a polar molecule or polar substance, so water sticks to it, and wherever water comes into contact with glass, it sort of climbs up the walls of that glass tube. And that creates this sort of cup shape in the top of the water column, which we call a meniscus. So this half circle is a meniscus. And so one thing that we'll see in lab potentially is that the narrower capillary tube is, or the more contact water has with the glass walls of a tube, the further up that water is going to climb. Right, so again, water is a polar molecule. You keep hearing me say that over and over and over again. Very important. The single most important thing in this entire chapter. It affects everything from solubility of substances to barriers around cell membranes to just about anything you can imagine in biology. So it's a very important polar molecule, right? And so again, what that means is that since we have a difference in electronegativity between oxygen and hydrogen, we create partially charged ends on the water molecules, right? So if we jump back here, we've got partial pluses. These would be your two hydrogens. Partial minus would be your oxygen. So that creates attraction between the partial minus on one molecule and the partial plus on the next molecule, partial plus on this molecule and the partial minus on the next molecule. There's a lot of attractions here. Those hydrogen bonds are very, very important, right? So I mentioned before, water transport. The reason why trees can be tall and transport water from the roots up to the top of the branches is because of water being a polar molecule, and also the polarity of the tubes, the water transport tubes that we call the xylem inside the stem of that plant. Right? Another factor of water that's important is that water has a very high specific heat. Specific heat is defined as the amount of energy it takes to increase the temperature of a substance by 1 degree Celsius. And so water is a very good substance which is very good at soaking up extra heat without changing in temperature. And so that's important, right? That means that since your body is made up of mostly water, your body has a certain amount of thermal inertia, whereas the environmental temperature around you might change. Because your water is made up of largely water, it's much harder for it to change temperature. Right? It also means that if you live near a body of water, the temperature, the climate's much more stable than if you live inland, because water can soak up heat in the summertime, release heat in the wintertime, and then it helps to stabilize the climate around where you live. So it's very important for that reason as well. Water has a very high heat of vaporization, which means it takes a lot of energy to transform water from a liquid state into a gaseous state. Why is this important? This is how sweating works, right? When water evaporates from your skin, it pulls heat energy away from your skin, thereby cooling down your body temperature. So that's also a very important factor. Solid water is less dense than liquid water. So you may want to think, about everyday life and how you might know that, but solid water basically means ice. Liquid water is liquid water, and so if you have ice in liquid water, what happens to it? It floats, right? And so because it floats, that means that solid form is less dense than the liquid form in which it's floating, which is really unusual for a substance. Most substances are more dense in the liquid state than they are the gaseous state, and so that's going to be a little bit different here than what we see with most other substances. In this case, the solid form is less dense, the liquid form is more dense, and that's backward for a lot of substances. So ice floats. And why is that important? Well, if you happen to be an aquatic or semi-aquatic organism in the wintertime, this is what keeps you from freezing solid. Because if the air is really, really cold, where does the ice form first? It forms on the surface, right? Because ice floats. And once that ice covers the surface of that body of water, it protects and insulates the liquid water beneath, thereby protecting the living organisms living in that water. So if you're a reptile, if you're an amphibian, a fish, an invertebrate, you're below the ice, you're insulated, it's not as cold, and the water's not going to freeze solid. If ice formed on the bottom of a surface or a body of water, if ice formed on the bottom of a body of water, ice would form in layers from the bottom up, and eventually what we'd end up with is one solid block of ice with animals frozen in the middle of it, and they would basically all die. Water is also a pretty decent solvent. Almost any sort of polar substance can be dissolved in water. So if you're looking at things that are ionically bound, like salt, if you're looking at things that are other polar molecules, like some proteins, like carbohydrates, those things are very able to dissolve in water. And so it's a pretty good solvent. Sometimes you hear water referred to as the universal solvent, which is not technically true 100% of the time, because a lot of hydrophobic things obviously don't dissolve in water. But since so many things are hydrophilic, water is seen as a very, very good solvent. And so again, hydrophobic means things that don't dissolve in water, water insoluble, versus hydrophilic things which are water soluble or water loving. And so we talked before in the previous section about like dissolves like. Polar dissolves polar, nonpolar dissolves nonpolar, polar and nonpolar not so much. Right? So we can see that with oil and water not mixing. If you have something like grease on your hands from your car, grease is a nonpolar substance. If you have grease on your hands and you just run your hands under water, nothing happens to the grease. It stays there, it doesn't go away, it doesn't wash off because it's not water soluble. So if you want to get grease off your hands, you have to use something which is also nonpolar, or at least partially nonpolar, so it can actually remove that substance. The last little section before we get into our biochemistry component of this chapter is on pH. And so as you move up into some of your higher classes in biology, you're going to see places where pH becomes incredibly important, especially as it pertains to homeostasis and the role that it plays in affecting various systems in the body. So pure water, pure deionized water with nothing else, no other contaminants in it, is called neutral. And so the reason why it's neutral is that it has a concentration of hydrogen ions in it that are 10 to the negative 7th moles per liter. Now that's a lot of stuff without a whole lot of explanation. Right? The H pluses are what actually makes something an acid. This means it's an acid. This is a measure of concentration. This number actually would be 0.0000001 moles per liter, and moles per liter is a measure of concentration. So anything with an exponent of 10 to the negative 6th or bigger is an acid. Anything with a concentration of 10 to the negative 8th or bigger, or actually, yeah, larger, is going to be a base. So if you see 10 to the negative 6th, acid. 10 to the negative 5th, acid. 10 to the negative 4th, it's an acid. If you see 10 to the negative 8th, negative 9th, negative 10th, those things would all be bases. Right? And so those can actually be converted into decimals, which makes it a little easier to see why the concentration of H pluses is higher at 10 to the negative 6th than what would be at, say, 10 to the negative 8th. Right? So water molecules in a pure state, pure deionized water, will break apart at a very fixed rate and produce both H pluses, which makes them an acid, and OH minuses, which makes them a base. And because they give up these at equal ratios or equal rates, that makes them effectively neutral. Okay, so H2O is a hydrogen ion plus a hydroxide ion. So therefore, if it breaks apart, it gives you your H pluses back and your OH minuses back, and it makes you neutral. Right? Anything that is very acidic or acidic is going to have a low pH. Anything that's not very acidic or that is very basic is going to have a higher pH. Right? So low pH means acid, high pH means base or alkaline. All right, so if we take a look at some acidic things, any sort of a substance where the concentration of H pluses is bigger than the concentration of OH minuses is an acid. Right? So for example, hydrochloric acid, which is what we produce in our stomachs, breaks apart to form H pluses and Cl minuses. And since it's producing H pluses, that makes it an acid. So HCl is a naturally produced acid. Acetic acid is naturally produced. Citric acid, we find that in fruits, is also a naturally produced acid. Formic acid, which is produced by ants, is a naturally produced acid. Acetic acid, by the way, is vinegar, so that's a natural one also. If you have a situation with a substance where the OH minuses are bigger than the H pluses, that would be a base. So sodium hydroxide, which is also known as lye, 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 breaks apart into Na pluses and OH minuses. So that makes this a base. Right? Now, these last two bullet points are going to seem to be fairly confusing the first time you see them if you've never looked at pH before, but we'll walk through this kind of slowly. Right? So the way we calculate pH is by looking at the negative logarithm of the concentration of H pluses in solution. And we'll explain what that means here in a second. Another way to think about this, because it is on a log scale, every step on the pH scale represents a factor of 10. So we'll look at some examples and see how the difference or the concentration difference in different substances varies as you become more basic or more acidic. Okay, so that's going to take a little bit of explanation. So if we take a look here at this pH scale, right? We've got a pH scale going from 7, which is neutral, acids being up here, bases being down here. Low numbers are acids, high numbers are bases. Okay, so if we take a look at the numbers over here on the left, 10 to the negative 6th, if you take the negative log of 10 to the negative 6th, you get 6. Effectively, what you're doing is bringing down the exponent, changing the sign, that gives you the pH. Negative log of 10 to the minus 10th is just 10. Negative log of 10 to the minus 3rd is 3. So it's actually fairly easy to see how this is calculated once you know what the numbers actually mean. Right? But because we're dealing with a log scale and because we have 10 to the minus 1, minus 2, minus 3, minus 4, and so on, every time we change one step on that pH scale, we're changing by a factor of 10. So if we go from coffee down here on number 5, pH 5, to orange juice at pH 3, that's a change in concentration of H pluses of 100. So this first step gives us 10. The second step gives us 10 more. So 10 times 10 is 100. If we go from soap up to pure water, we have 10 times 10 times 10, which is 1,000. So water is 1,000 times more acidic than soap. If we were to go with seawater versus tomato juice, we have 10 times 10 times 10 times 10, which is 10,000. So tomato juice is 10,000 times more acidic than seawater. Every step on the scale is a factor of 10. So every time you change one step, you have to multiply by an additional factor of 10. If you go the other way with it, if we say, okay, we're up here at lemon juice, and we're going to look at how that compares to coffee, well, now we're going to say that coffee has, let's see, one step, 2, 3. Coffee has 1/1000th, or 1 over 1,000 concentration of H pluses as compared to lemon juice. Okay, so these guys up here have more H pluses than these guys down here have. And so when we relate pH values, we have to say which one's more acidic and how much more acidic it is or how much more basic it is. And so that's how this pH scale kind of works. And it takes practice, right? So we're going to work with this in lab. We'll see some examples of this, and it will eventually make more sense. But the key is to remember what pH actually represents, and that's concentration of H pluses, and then how that concentration is related to the scale itself, and that's where the negative logarithm comes in to play when it comes to calculating the actual pH from the concentration of H pluses itself. Now, buffers are things which have the ability to soak up or give off H pluses as acids or as bases are added to them. Right? So they resist changes in pH. So if you have a substance, it's a buffer, and you add a base to it, that buffer will actually release extra H pluses to neutralize the OH minuses that are showing up in that base. So by doing that, the H pluses join up with the OH minuses, they neutralize it, they create water, and that keeps your pH from changing. On the other hand, if you have a buffer and you add an acid to it, it's going to give off OH minuses. It's going to soak up the H pluses that you are releasing, and that's going to allow you to keep, again, the pH at a relatively consistent level. So these are really good at maintaining homeostasis of pH in the body. And the reason why that's important is because pH has the potential to affect all kinds of factors and conditions in our bodies, especially when it comes to proteins. One of the environmental factors that proteins are most sensitive to is pH. And if you acidify them or make them more alkaline, those proteins are very likely to come apart and stop working. And if those proteins happen to be enzymes, that means a whole bunch of chemical reactions in your body have the potential to stop. And that's going to lead very quickly to you ceasing to exist anymore. It's going to be a very bad situation if those enzymes fall apart and fail to function. Right, so here's an example of a buffering situation where we have pH going from 0, very, very acidic, to 9, which is fairly alkaline or basic. This particular buffer is active within a range of around pH 4 to pH 6. All right, so we start off at pH 2, we're an acid, we're going to add in some base to it. And as we do that, the pH goes from 2 all the way up to about 4. It's a really big drastic change in pH. It's changing by 100 times. So in other words, we're saying that now the substance is 1/100th the acidity as it was before we started this little project. Okay, so we're going to change quite a bit from 0 to some amount. So we'll say we're adding 100 mils. We had 100 mils of buffer to this, and the pH changes by 100 times during that 100 mil addition of base. Now the buffer is really active between pH 4 and pH 6. So once we're at 100 mils, let's add another 100 milliliters into the situation. Now we've got 200 mils, but look how the pH has changed. It went from a pH of 4 to a pH of around maybe 5 at most, maybe 4.5. It's not that big of a change, probably more like 4.5. And if we add another 100 mils, that takes us to 300 total. That's going to give us a pH of 5. And if we add another 100 mils, that takes us to a pH of 5.5. So we've just added in, we basically added in 300 mils more, and the pH is only changed by 100 times. Or the pH scale is changed by 2. It's 100 times the concentration. So that's a relatively small change. The buffer is doing a really good job of soaking up those OH minuses by releasing H pluses and keeping the pH at a relatively consistent level. If we didn't have the buffer there, the pH would have gone through the roof back here at the 200 mil, 300 mil, or 400 mil type range. Now, the buffer's only got so much room to work, and if it gets to be too big of an addition of base, then eventually the buffer runs out of H pluses to offer up. So as we go from 400 to 500, all of a sudden the pH goes through the roof because you've basically exhausted the capacity of buffer to supply H pluses to the solution. But for a while, it did a pretty good job of keeping us within that very, very narrow range of pH. All right, so one application of this is to look at what's called the blood buffering system that we have in our own bodies. And we're not going to hold you responsible for this for right now, but this is something you'll see as you move up through, say, anatomy and physiology, is how this process works. Right? So it turns out that if you take bicarbonate ions and hook them together with hydrogen ions, you can make what's called carbonic acid, especially if you have a certain kind of enzyme present to make that happen. Carbonic acid is formed. Now, carbonic acid can break apart to form bicarbonate and water in the presence of hydroxide ions. So it also has the ability to break apart. So what this does then is it creates a potential for this substance to produce H pluses in some situations or produce OH minuses in other situations to help neutralize pH. If pH in the blood, say for example, is becoming too basic, this type of situation can happen. You create carbonic acid, that makes the pH come back down again. If the blood's becoming too acidic, you can then produce OH minuses, which is then going to help eventually neutralize the blood and bring the pH back up again. So it's a very nice automated way we have of keeping the pH within a relatively narrow range. All right, so with that, I'm going to cut off this section, and when I start up the next section, which should be section 5, we're going to start looking at organic molecules, how they're built, how they're taken apart, and the different kinds of organic molecules and what they do.